gentlemen, my name is Lung Prasad, and this summer I had the honor and the privilege of working under the mentorship of Dr. Shilpa Chatterjee with the assistance of Jian Chen Tao and Elena Sorokina in the Department of Physiology at the University of Pennsylvania Perelman School of Medicine on a project entitled IR Injury and Lung Transplantation, ROS-Induced CAMS Amplify Injury. Before I delve into the specifics of my research, I will start with a little background. Lung transplantation, also known as pulmonary transplantation, is a routine clinical practice in the treatment of chronic organ failure and end-stage disease. A recipient's infected lung is removed from his or her body, and the donor's appropriate lung replaces it. Therefore, there are three essential stages of lung transplantation: removal, storage, and reattachment. During transplantation, organ removal from the donor includes a stage when the blood flow to the organ is stopped. Following organ removal, the storage of the organ lasts for 3 to 6 hours. Finally, the blood flow is restored with the organ, the blood flow is restored with the recipient's blood at the time of transplantation. The temporary interruption of blood supply to the recipient's organ, which happens during the storing phase of transplantation, is called ischemia. On the other hand, the resurgence or restart of blood flow is called reperfusion. Ischemia-reperfusion injury, also known as IR injury, plays an important role in organ transplant failure. During the stage of ischemia, there's an absence of oxygen and nutrients, which leads to the production of ROS, also known as reactive oxygen species. ROS is known to cause oxidative damage and inflammation. Although the problem with the lack of oxygen is solved when the donor's lungs is kept inflated, the loss of the mechanical component of flow triggers the signaling that produces ROS. To improve the management and outcome of transplants, we need to understand the various steps that lead to oxidative damage and inflammation with IR. Consequently, there are 3 stages of ischemia-reperfusion injury: ischemia, ischemia storage, and reperfusion. White blood cells protect the body from infections and diseases. Polymorphonuclear neutrophils, also known as PMNs, are the most abundant type of white blood cells in mammals, and they play an integral role in the body's defense mechanism against pathogens. They are included in a group of white blood cells called granulocytes. Neutrophils are sent to the site of injury, in this case the IR injury, where they generate superoxide anions throughout the NADPH oxidase pathway. The NADPH oxidase pathway is a membrane-bound organelle complex, which is used by neutrophils in the plasma membrane. Since we do not know the intermediate steps, our goal through this experiment is to learn them. The superoxide anions, which are created via the NADPH pathway, can form hydrogen peroxide, which further yields reactive oxygen species. The production of ROS happens in the mitochondria as a part of everyday aerobic life, and they are also produced in enzyme reactions. ROS is necessary for life, but ROS in excess can be very detrimental. While ROS can act as a pathogen, in this case it is very harmful to the cell. ROS does damage to the cell in 3 distinct ways. It oxidizes fatty and amino acid, it inactivates enzymes by oxidizing cofactors, effectively breaking down these cellular machines, and it damages DNA. One of the main ways ROS can do damage to the cell is by causing oxidative stress. According to the National Institute of Health, oxidative stress is the disturbance between the production of ROS and antioxidant defenses. Oxidative stress can lead to acute respiratory distress syndrome, also known as ARDS, and chronic obstructive pulmonary disease, also known as COPD. The 3 main situations in which ROS are formed are neutrophils, cells exposed to abnormal experiments, and ionizing radiation. This figure shows everything that makes up reactive oxygen species. These are the 3 parts that make up lung transplantation. There's stop, storage, and restart. All of these have one thing in common: they produce ROS. ROS further yields oxidative damage, and oxidative damage through the immune response amplifies IR injury. We question why ROS leads to the amplification of IR injury. If we can block ROS, then we will be able to stop oxidative damage and lessen IR injury. This leads me to my research design. My purpose for this experiment was to understand the intermediate events that lead to inflammation in a model of ischemia and reperfusion. I hypothesized that reactive oxygen species produced with ischemia will generate increasing adhesion molecules through the activation of transcription factors. These adhesion molecules recruit more immune cells, which will produce more ROS, and which will further amplify IR injury. We used two animal models of lung transplantation. First, we used the mice, and then the pigs. In a previous experiment, my lab did an identical test on mice lung tissue, and the methodology was stated as: the mice were prepared for transplantation. Next, the mice were clamped to imitate IR injury. Finally, the mice lungs were transplanted. On the other hand, my methodology, which was used with the pigs, was stated as: the slides were washed, sectioned, and incubated and stained with the primary antibodies. Then the slides were washed and stained with the secondary antibody. Following that, the slides were mounted and prepared for viewing. And finally, the slides were viewed under a microscope and analyzed. These are my materials. Please take a moment to look over them. First, the slides were washed with phosphate buffered saline, also known as PBS, 3 times and left for 5 minutes each time. Following the wash, they were incubated with 0.5% trident and PBS, and then rested for 20 minutes on ice. Following the wash, they were incubated with they were washed again with PBS 2 times and left for 5 minutes each time. Following that, they were incubated with 0.3% glycine and PBS for 30 minutes. Then they were washed again with PBS for 2 times and left for 5 minutes each time. The slides were diluted in 0.25% DSA and 0.25% NGS and 0.01% trident and PBS for 1 hour. To conclude that day, the 2 antibodies were added to the tissue on the slide and left overnight. The next day, the slides were washed again with PBS 2 times and left for 5 minutes each time. Next, the secondary antibody was added. And to complete the preparation process, the slides were washed first with PBS 2 times and left for 5 minutes each time, and next with DH2O 2 times and left for 5 minutes each time. The slides were then mounted with cover slips and 1 drop of vector mounting medium with DAPI, and left in the cold room overnight in the dark. We used all of our 12 slides were viewed through the technique of fluorescence microscopy. We utilized this technique to localize specific proteins and other subcellular structures within the cells. While we only utilized one aspect of fluorescence microscopy, there are many others that can be used. In the figure to the left, it shows the microscope that we used, while on the right, it shows the image of the entire room, where this is the microscope, this is the source, and this is the computer we used to get the imaging. This leads me to my results. This is how an adhesion molecule affects the lung tissue. The neutrophil is captured and begins to stick with the cams. Then it rolls and proceeds to arrest and then continues. The 2 adhesion molecules that we studied were ICAM and VCAM. While both of them were adhesion molecules, one was intracellular and the other vascular. Both recruited neutrophils to stick and bind to the cell surface. As I mentioned before, in the previous experiment, my lab did an identical test on mice lung tissue. And on this test, the same trend of increase was shown. The blue showed the original cells, while the red represent the ICAM. There are 2 types of cells shown here: wild type, which are original, and NOX2 null, which are cells without ROS. Each of the 2 cells are divided by 3 sections each: 1 unligated, 2 ischemia, and 3 IR. In this case, in the wild type cells, there's a clear increase in the ischemia and reperfusion stages, while in the NOX2 null stages, there's little to no ICAMs present. As I mentioned in the previous slide, there are 2 different types of slides divided by 3 sections each, except in this figure, the green represents the VCAM. Just like in the slide before, in the wild type slides, there's increase in the ischemia and reperfusion stages, while in the NOX2 null stages, there's very little increase in VCAM shown. The pig donor lungs were stored for 2 hours and then transplanted into the recipient pig. Unlike in the mice where wild type and NOX2 null were compared, in this case, for the pigs, VCAM and sham, which is original, and in post-transplant were compared. In the post-transplant tissues, there's a clear increase in the IR stages, while in the sham slides, there's very, very, very little ICAM shown. As in the previous slides, there are two different types of slides divided by 3 sections each. In this case, the red represents the VCAM. In the post-transplant tissues, there is an increase in the reperfusion stages, while in the sham, there is no VCAM shown whatsoever. This leads me to my discussion. Before the experiment, I hypothesized that reactive oxygen species produced with ischemia will generate increasing adhesion molecules through the activation of transcription factors. These adhesion molecules recruit more immune cells, which will produce more ROS, which will further amplify IR injury. My hypothesis was accepted due to the fact that the adhesion molecules increased in both the ischemia and reperfusion stages, while in the NOx2 null slides and the sham slides, there was no increase whatsoever. For future research, we would like to continue research on IR, assessing different compounds in ischemia and reperfusion, and begin human trials. If we are successful in our future experiments, we will be able to begin human trials in 5 to 6 years. These are my acknowledgements. Please take a moment to look over them. Thank you. Any questions, comments, or constructive criticism?